fall and are starting things on a high note with Dr. Joseph Hill. As a reminder, for those of you who would like to view Research Conference but can't attend in person, we do live stream, and that link's on the DCRI website, and we also archive every session on our YouTube channel about a week after the session. So make sure to keep an eye out for that if you're out of town and unable to attend. So now I'll turn things over to Dr. Oman.
Thank you, Emily. I'm Magnus Oman, and it's great to be back here for DCRI Grand Rounds. We're very pleased to have an old friend, but also somebody who's contributed significantly to science in cardiovascular medicine in the United States and worldwide in Professor Joe Hill. Joe actually has a local connection. He went to high school in Burlington, I learned. And so for those of you who are native North Carolinians, and there are not that many at DCRI, but for those of you, you will recognize the connection. And then, so he is also an undergrad at Duke and did a doctorate in pharmacology here at Duke before taking a 5-year sabbatical. For those of you who are residents or fellows, 5-year sabbaticals in Paris are highly recommended if you look at Joe's distinguished career. So Joe is also the editor-in-chief for Circulation, a fairly well-known medical journal, and the official AHA Journal of Medicine or Journal of Cardiology. So, Joe, it's great to have you here. And as it would be, he also has a child at Duke, but that child decides to do a term abroad this term. So he doesn't get to see him, but at least it's great to have you here. Joe, welcome.
This thing on it. It's an absolute pleasure to be here. I have many fond memories of this place, and you'll be happy to know I write checks every 6 months to Duke. So, so I have a special relationship now with Duke. But we will, in 2 years, we'll have 5 Duke degrees in our family, and you know, so it's an absolute pleasure to be here. I haven't had occasion to be here for an extended visit for a while, and so this is absolutely a wonderful day for me. And Magnus decided, since we're launching your new year of seminars, that he'd start you off with some vegetables, and that is basic science, something that's it's good for you. Trust me, this is going to be good for you, okay? And then later on it'll be dessert and more pleasant stuff. But if you think about the diseases that kill human beings in this world, they are, number one, you know what, I've got a bright one right here. Blood pressure. No, I don't. Okay, I did it. Blood pressure. Blood pressure is the number one killer in this world. And that is the first disease that I started to study when I launched my own lab. This is not very good. Some years ago, and I'm going to tell you something about that. The number 2 killer is, if you can think of this potentially as being ischemic heart disease, tobacco, cholesterol, those sorts of things, ischemic heart disease, and we're going to talk about that. And if you jump over the things that challenge the developing world, like malnourishment, the 3rd killer in this world, this is just not a happy pointer right now, is diabetes. So I'm going to tell you 3 stories about the 3 killers in this world. And I'm going to argue for you that ischemic heart disease is actually falling down the list and diabetes is climbing up the list. So what I'd like to do is tell you stories about these 3 things, which are things that we've worked on in my lab now for some years, that contribute in aggregate to a huge burden of cardiovascular disease around the world. These are the environmental influences that impinge on the heart that ultimately result in heart disease. So all of heart disease is a result of this exposome, where the heart is exposed to elevated pressure, like high blood pressure, elevated volumes if you have a defective valve and so forth, neurohumoral stress, ischemia, reoxygenation, and probably the most recent one that is rapidly expanding is metabolic stress. So I'm going to start off by telling you a story about pressure, stress, hypertension, one about ischemia reperfusion stress, and one about metabolic stress. Again, these are the three biggest killers of human beings on the planet. The take I'm going to put on this is an epigenetic one. Epigenetics, as you know, is the control of gene expression in the absence of changes in DNA sequence. So there are many ways in which a gene is controlled above and beyond changes in its promoter and enhancers and so forth. Those epigenetic mechanisms are governed by three different classes of events. One are writers, which are proteins. This pointer is just not having a good day. Writers, which are molecules or are enzymes that confer either methyl groups, acetyl groups, or phosphate groups onto either DNA or the proteins around DNA called histones. So DNA is wrapped around these proteins called histones, and these post-translational modifications are conferred by these enzymes, which either put a methyl group in places, an acetyl group, or a phosphate group. Those writers are balanced by erasers that take the acetyl group off, histone deacetylases, or take the methyl groups off, writers and erasers, and there are readers, proteins that read these epigenetic marks and govern gene expression. That's the fundamental essence of epigenetic gene control. I'm going to talk mostly about histone deacetylases. These are the enzymes that strip acetyl groups off of histones. And the reason for that is because these molecules, I'll show you, govern a great deal of this biology. And also, there are small molecules, in other words, drugs that people take, FDA approved, to alter histone deacetylase activity in several rather rare cancers. Also, let me emphasize while I'm on it that histone deacetylases deacetylate histones. That's where they were discovered. They also deacetylate about 1,000 other proteins. So the H in HDAC is a misnomer nowadays, is an anachronism really, because there are many, many proteins that are deacetylated by HDACs even that are not histones. So even though I'm illustrating this at the level of histone, the closure and the opening of chromatin, I'd like to emphasize that histone deacetylases deacetylate many other proteins. So over the course of the next 45 minutes or so, I'm going to build a model. And the first, where we relate cellular processes and cardiovascular disease. The first one, I think everyone is probably pretty well convinced right from the start that hypertrophy is related to pressure overload. In other words, elevated blood pressure is associated with hypertrophy of the heart. So that's our starting point for the model that we'll build over the next several minutes. So we'll just tuck this right over here on the side. And I think we all can accept that hypertrophy does in fact contribute to the biology of pressure overload stress. And I hope you all will recognize that there's this molecule that will be central to my talk here, mTOR, mechanistic target of rapamycin, which is a master cellular regulator of cell growth. So we've known now for well more than a decade that mTOR is a master regulator of cell growth and contributes importantly to the disease of hypertension. Let me introduce a new concept to you, and that is autophagy. I don't know how familiar you are with autophagy, autophagy, self-eating. All cells in your body cannibalize themselves a little bit constantly all the time. Cardiomyocytes do that as well, all the time. Every cell in your body undergoes this catabolic event where cytoplasmic material is engulfed in a membrane-enclosed organelle, which fuses with a lysosome and dumps in acid hydrolases and degrades all that captured protein and captured nucleic acid and lipids and so forth and releases into the cell. Every cell in your body is doing this right now, and if you were to squash it, eliminate it, that cell would die over the course of several hours. If you take a yeast cell and you prevent it and you starve it, for example, what does it do? It starts to nibble away at itself even more to maintain cellular homeostasis. If you starve a cardiac myocyte, the same thing. It nibbles away at itself to release building blocks for survival. If you take a yeast cell and you prevent that from happening, the yeast cell will die within the course of a few hours. As a dramatic example of the importance of autophagy, is a little digression here, in mammalian biology, if you think of a mammalian fetus in the womb is being fed with nutrients and oxygen 24/7. from the moment of conception to the moment of birth, that creature, that little baby or that little mouse pup, is eating 24/7. Then comes birth, and for the first time in that creature's life, it is now starving for several hours, an experience it has never had before. During that process, when the baby and mom are trying to figure out how things work, that fetus is starving, and autophagy lights up across the entire organism to keep that little baby alive. If you engineer mice where you prevent that from happening, such that when they're born, they cannot mount an autophagic response, the mortality at 12 hours is 100%. So mammalians cannot survive the birth experience without this autophagic response. Now, in terms of cardiology, I will tell you that I have never seen a situation in either preclinical or clinical disease-related context where the myocyte does not undergo a shift in its autophagic flux response. Often it usually goes up. Often it goes up and comes back down again. Sometimes it goes down and stays down. But it is absolutely a universal thing, and we've been working on this for more than a decade now. And one of the puzzles we have worked on and are still working on is trying to understand whether this autophagic response is a way in which the cell is trying to protect itself from these stresses in order to prevent disease progression, or could it be that this autophagic response is actually conferring harm and promoting disease pathogenesis? I will tell you the answer is it depends. It depends on the context and the type of stress. Usually the autophagic response is a good thing, something we would want to promote, but there are definitely instances where it becomes maladaptive. So we're going to talk a little bit about autophagy now that I've introduced it. So let me talk about, first of all, pressure overload. The way we do this preclinically is by putting a surgical constriction on the aorta of an animal, thoracic aortic constriction, thereby raising afterload by about 70 or 80 millimeters of mercury. And when you do that, the growth of the heart weight normalized to body weight goes up. In the next 3 weeks, the heart hypertrophies because it's having to work extra hard. It hypertrophies around 40% or something like that. If you take, these are ancient data, now 10 years old or more. If you take animals and you expose them to a compound called trichostatin A, which is a broad spectrum HDAC inhibitor, it basically silences many different HDACs. Under resting conditions, nothing happens, the heart is normal, and the heart will grow under the non-drugged conditions. But in the presence of this compound, the heart growth is essentially cut in half, suggesting that about half of the heart growth response is HDAC dependent and another half is not. If you look at just the LV alone, you see essentially the same thing, that some element of the growth response involves these HDACs that I've told you about, and those mean data are corroborated here structurally. So this is ancient data that we've shown now for a long time that, as I told you, that if you stress the heart, autophagy changes, and it turns out that if you block that autophagic, if you block the HDACs, that growth response is blunted, and in fact that involves autophagy. So if you'll take my word for it, that the abundance of this molecule right here, quantified here, is a snapshot of the number of these organelles called autophagosomes that are trafficking through the cell. So the more of this band here, the more autophagosomes you have in your cell. So as I told you, if you expose the cell to most anything, including intact, autophagy goes right up. And so Diane Cow, a fellow in the lab, asked the question, what if we blunt heart growth, which I just showed you that TSA can blunt it by about 50%. Turns out the autophagic response is similarly blunted. Now, this doesn't tell you which is the chicken and the egg. Did the heart not grow because there's less autophagy, or is there less autophagy because the heart didn't grow? I don't have time to show you this, but it is the former, that by blocking the autophagic response, the heart can't grow. I've told you that autophagy occurs in virtually every context. It turns out, like many things in the myocyte, which is a rather remarkable thing, is if you prevent it from having that response, it doesn't respond well, and the plasticity response is completely aborted. As in a digression, the heart eats about 70% of its energy from lipids and about 30% from glucose. And under conditions like this, that shift goes more toward using glucose more toward a 50-50 split. If you prevent that, if you clamp it at 70-30, the heart won't grow either. So this plasticity of the myocardium is intimately involved, or requires an autophagic response and also metabolic and other events. So Diane, who is a cardiologist, thought, okay, we have this small molecule that will clearly alter the growth response of the heart, and it seems to do so by means of, the way in which it does that is through autophagy. So she engineered mice that express a molecule called Beclin-1. But if you'll take my word for it, that Beclin-1 is a protein that's involved in basically amping up the autophagic flux response. So if you put extra copies of Beclin-1 here exclusively in cardiac myocytes, the heart is normal under resting conditions. You haven't changed anything, but under conditions of stress, this autophagic response is amplified about threefold, it turns out. And so she thought, if these HDAC inhibitors have any potential to be useful in a translational sense, and given that we know that they work through autophagy, let's make their job as hard as possible by amplifying that autophagic response to see if they still work. So with these animals overexpressing Beclin-1, under conditions of TAC, the heart grew quite a bit. Under conditions of TAC for 10 weeks, the hearts were absolutely enormous. And so in parallel, she did this same experiment where she exposed animals to that HDAC blocking drug, TSA, to see what would happen. And what she found was that growth response was remarkably attenuated. So those data are quantified here, where the growth response that we normally would see is amplified if the animals express Beclin-1. In other words, this increment here stems from the amplified autophagic flux event in those cardiac myocytes. And if you take either one of them and expose them to an HDAC inhibitor, boom, they come right back down to the same level. And this increment that is autophagy dependent is abolished. And again, as we had seen some years previously, there's an element of growth that persists and HDAC independent. So I'm telescoping a lot of data here, but Diane, who is a cardiologist, obviously recognizes that even though we try to prevent disease, often we're unable to do that. Patients often present at the time of symptoms with structural heart disease. So in addition to the prevention experiment that I just showed you, she also asked the question, is it possible to reverse some of this? So she took these same animals that express extra Beclin and hence extra autophagy, and under resting conditions, the mass of a mouse LV is around 65 milligrams, that's normal, and percent fractional shortening by echo is around 70. So that's a normal mouse heart. After 10 weeks of TAC, the heart undergoes a dramatic threefold growth response, and LV function declines. So this is when the patient walks in your clinic with LVH, systolic dysfunction and symptoms. What happens if you then expose them to TSA with the constriction still in place? The surgical constriction is still in place. She found that over the next 4 weeks, LV mass began to return back toward normal, LV function began to improve, and in fact, LV mass continued to decline yet further with an additional 4 weeks of treatment, and LV function returned essentially back to normal. It's quite an amazing plasticity response. We believe that this increment of growth that has persisted here is probably extracellular matrix and fibrosis that also is quite plastic, but with much slower kinetics. So back to our model. I hope I've convinced you now that autophagy plays a role in pressure overload, the #1 killer in the world. And if you'll take my word for it, that mTOR is known to be a master inhibitor of autophagy. So we're starting to see that now focusing on this one disease here, we have two events that are both of which are governed by mTOR. mTOR controls heart cell growth, and mTOR suppresses this catabolic autophagic response. Second type of disease, ischemic heart disease. This poor gentleman just stepped out of a restaurant here in Boston, and you can see he dropped his briefcase and dropped his cigarette right there, and is suffering a myocardial infarction. So we're going to talk now about ischemia and reoxygenation stress. So here he is on the street. He has a coronary artery occluded, and he's losing myocardial mass every minute that he's standing there. He makes his way to the emergency room where they give him nitrates and morphine and a beta blocker and an ACE inhibitor, and they take him to the cath lab and open up his coronary artery at this moment, thereby saving coronary myocardial tissue, PCI salvaged myocardium. That is the state of the art in 2016 around the world for dealing with a myocardial infarction or with a stroke, as all of you know. It's important to remember, though, that the injury response is not abolished at this point, but rather is slowed. There is an ongoing injury response, and this component here is called reperfusion injury. If you have a heart attack, half of the injury is ischemic, and half of the injury is reperfusion related. The way we treat heart attacks these days is to diminish the ischemic burden by lowering stroke work and heart rate and delivering oxygen and so forth, to try and improve this component here, and of course opening the coronary artery. The number of therapies that we have to treat the other half of the problem equals 0. 0. There are no therapies whatsoever to target half of this problem. Same in a stroke, where nowadays the neurologists do carotid angioplasties as well. We have 0 therapies targeting half the problem. That's amazing, right? And it's interesting, if you go back and look and ask the question, why is that? Why is it that 30 years of work in the preclinical space has never translated into something that has been clinically useful? I think there are many possible explanations, and of course, perhaps one of the most concerning one is that taking a normal coronary artery in an animal and wrapping a suture around it and then releasing it is not the same thing as a thrombosed, inflamed plaque in a patient with diabetes. Those are just not the same biology. So maybe that is part or even all of the problem. But I think it probably is more than that, because there are many instances where people who do this kind of work like me over the years have not used the kind of rigor that we would all expect nowadays to impose here. For example, you wouldn't launch a clinical trial nowadays here in the DCRI based on results from one lab or one animal model. You wouldn't launch it if the signal were not robust, if there were only a little bit of protection. You'd not do that. You'd be surprised how many times that has happened in the past. Perhaps the most concerning of all is that many of these trials in the past have been launched in the setting of pre-treating the animal with drug and then injuring him and noting protection. That's like treating the patient on Monday because you know he's going to have a heart attack on Tuesday. That has no clinical relevance. So it's actually shocking if you look back the number of times in which the preclinical biology was really not up to a standard that one would expect in this space. So recognizing in our lab that the enormity of this problem and that there's still room, I think, to do the science better, we got involved with this. And to make a long story short, again, I'm telescoping a lot of work. We started off in mice, and we found that we could confer 40% cardioprotection in a mouse if you delivered a compound, an HDAC inhibitor, at the time of reperfusion, when you open up the artery. 40% is a big signal. It occurred at the time of clinical relevance, in other words, reperfusion. That observation was made in 2 other labs around the world, so it was reproducible across independent labs. And with that in mind, we then moved on to a large animal trial in rabbits here, where we figured out a way to reversibly ligate the circumflex coronary artery in these animals. And so using the same rigor that one would use in a human clinical trial, we randomize animals at this point, 24 hours later, expose them to 30 minutes of ischemia, then we reperfuse another 24 hours, we do echoes, and we sacrifice them. We randomize animals into 3 arms. In one arm, they received a dose of drug here, a second dose, a third dose just before surgery, and a 4th dose at the time of reperfusion. That would be the pre-treatment arm. In the other arm, we gave the animals a dose of vehicle, another dose of vehicle, and a single double dose of drug at the time of reperfusion. That would be the clinically relevant arm. And in the third arm, we gave them vehicle throughout. So all the animals got the same number of injections. It's only one of them got a clinically relevant paradigm, and the other was a pre-treatment paradigm. No one knew, just like the way you folks do your science, no, we didn't break the randomization code until the study was completely over, and we broke it and to find the results. And what we found was, in the animals that were pre-treated with drug, there was a dose-responsive decrease in infarct size, despite the fact that the surgeries were all the same. So in other words, we saw in rabbits exactly what we and others had seen in rodent models, that if you pre-treat the animals with drug and give them a heart attack, that heart attack is smaller. Those structural data are corroborated here functionally, where you can see that the decline in cardiac performance was attenuated in a dose-responsive way when you expose them to drug before injury. So now what about when you treat them at the time of the clinically relevant moment of reperfusion? There we found the same thing, that even when the drug was delivered at reperfusion, this similar decrease in infarct size despite the similarity of surgery And by echo, looking at either percent fractional shortening or ejection fraction, you can see that the decline in contractile performance was attenuated in both instances. Just briefly, I'll try to convince you that, first of all, if expose cells in culture to this HDAC inhibitor, it turns out the autophagic flux response that occurs in the setting of ischemia reperfusion is blunted in the setting of SAHA. It's complicated because what happens in ischemia, what happens in reperfusion are different. But I'd like to try to convince you that the HDAC inhibitors are also working through this autophagic response. And in fact, if you knock out autophagy using siRNA to silence several proteins that are critical to the autophagic response, that response, and I'm going through this quickly, is attenuated. So if you'll take my word for it, that the autophagy response is in fact critical to this IR response. So I've spent the last several years trying to convince several drug companies to do a clinical trial using an FDA-approved HDAC inhibitor in reperfusion injury. And I'm sure you know reperfusion injury is not a happy term in the pharmaceutical industry. So that has, and for that and also intellectual property reasons, it hasn't gone well. So actually now I'm trying to do this in China, where the number of infarcts in China is skyrocketing, whereas, as you know well, in the U.S., it's gone down. So it turns out this molecule is not CFDA approved in China, and so we're working on that to try to move this forward. So finally, let me tell you the third stress, and that is metabolic stress. So diabetes, in many ways, I think, is the future of cardiovascular disease. It's absolutely exploding on our planet. You can see right now it's estimated that there are 400 million people with diabetes already, and that number is expected to increase in the range of 500 million or more by 2030. And interestingly, you can see much of this is occurring in Asia. The prevalence of diabetes in the US is 11%. The prevalence of diabetes in China is 11%. The number of people in China, a country of 1.6 billion people with pre-diabetes, is 50%, 50%. There's going to be an explosion of diabetes and diabetic heart disease in the West, and that's one of the reasons circulation is, we're in China in a robust way, trying to get in front of that as best we can. So you may know that patients with diabetes typically succumb to heart disease, and if you think of it this way, there are 3 hits to your heart in the setting of diabetes. Many people with diabetes have ischemia, have atherosclerotic disease. Not all of them, but many of them do. Most of them are hypertensive, so that's the second hit. But the third hit is that the diabetic milieu itself is toxic to many cells, including the myocyte. If you think about fluctuations in glucose and insulin and a variety of cytokines and chemokines, adrenergic activation, RAS activation, the myocyte is being impounded with many, many stressful signals above and beyond the presence or not of ischemic disease and hypertensive disease. And that is called it's diabetic cardiomyopathy, a ventricular dysfunction that is above and beyond the presence of coronary disease and hypertension. So we set out some years ago to tackle this, again, being driven by the enormity of this problem and its expansion. And sadly, this is the underlying cause behind this. I have to tell you that the way in which our species is evolving. And now, as someone who has lived in Texas for more than a decade, I will tell you that we do this in Texas pretty well. So let me illustrate this. I like to do this with students sometimes. So this is 1985. This is a prevalence of obesity, not overweight, but obesity, okay? BMI greater than 30. For someone who's 5'4", that's 30 pounds. So in 1985, this is what we knew about the prevalence of obesity in the United States. You can see there are many states where we had no data. Several states where obesity rates were less than 10%. And a few states where obesity rates were 10 to 14%. That was 1985. 86, 87, 88, 89, 90. By 1990, we have data in most states, and you can see that many of the states have filled in with less than 10%, but now there are quite a few that have emerged with obesity rates in the 10 to 14% rate over 5 years. 91, 92, 93, 94, 95. By 1995, 10 years into it, there are no more states with obesity rates less than 10. In the West, there are a number with 10 to 14, and here in the middle, there are quite a few now that have emerged with 15 to 19% obesity rates, just in the course of 10 years. 96, 97, 98, 99, 2000. By 2000, we have no states with less than 10%. We have one lonely state, Colorado, with 10 to 14. We have a number of states here in the West with 15 to 19, and now in the middle has emerged unbelievably, just in the course of 15 years, a number of states with obesity rates that are 20 to 24%. 2001, 2002, 2003, 2004, 2005. By 2005, there's no light blue, there's no medium blue. There are only 3 states with 15 to 19%, and now we see these states with obesity rates 20 to 24%, 25 to 29, and even some that now have emerged unbelievably with obesity rates greater than 30%. Now we could stop there, or we could do 5 more years. 2006, 2007, 2008, 2009, 2010. These are the last data I have. There's no light blue, there's no medium blue, there's no dark blue. There are several states in this range here, but you can see that there's a huge swath of our nation where obesity rates have absolutely exploded. The face of our nation and cardiovascular disease has changed over the last couple of decades. And I will tell you that even though for both men and women in North America between 1980 and 2008, you can see obesity rates have dramatically increased. It's not just us. It's not just in the developed world. It's not just in the West. It is, in fact, a universal phenomenon where, in many ways, the shape, the features of our species has changed in the last 20 years. And you probably read the paper in JAMA a month ago, where the declines in cardiovascular mortality, actually in the last 5 years, for the first time in decades, flattened out. And many people think that's because we're going to start to see an upturn related to this here. But between obesity and diabetes and heart disease, there's a couple of decades or something of delay in there. And that maybe this flattening out in the last 5 years might be something related to this that we're seeing coming down the road. My friend and mentor, Dr. Braunwald, said the thrombocardiologist of the 20th century will be replaced by the diabetocardiologist of the 21st. So with that in mind, the magnitude of this problem, we got involved in this now some years ago, 10 years ago, and I'm going to tell you, here comes some more basic science. So that insulin, when it binds to a cell, it binds to the insulin receptor, and which in turn signals to the insulin receptor substrate, signals to a molecule called PI3 kinase and AKT, and sends a glucose transporter up to the cell surface. When insulin knocks on the door, the cell responds by sending a molecule to the surface to bring in glucose. At the same time, a molecule called FOXO, forkhead box O transcription factor, is inhibited. That is something that was known, and we set out to see if FOXO might have a role in heart disease, something that had never been explored before. FOXO is expressed at high levels in cardiac myocytes. No one had ever looked to see what it does. And it was known in other cell types that it had some role, like in hepatocytes, it had some role in diabetes. So we set out to answer that. And in fact, in work that I'm not going to really show you, we found that if you take FOXO and you mutate it so it's in the nucleus and on all the time, can't come out, stuck there, it makes the cell diabetic. It breaks the insulin signaling cascade by clamping AKT at a level of phosphorylation. So AKT can no longer hear insulin because it's stuck at this level of phosphorylation. So in other words, in the highly artificial context of putting FOXO in a heart cell and trafficking it to the nucleus and making it turn on, that cell becomes diabetic. So we then asked the question, does that have any relevance to a preclinical model of diabetes? For example, animals fed a high-fat diet, or animals that are mutated in their leptin receptor, 2 models of type 2 diabetes. And we found that in animals on a high-fat diet with diabetes, there are extra copies of FOXO in the nucleus relative to what one might have expected. Same thing in the DB-DB model. In these 2 independent models, FOXO is somehow in the nucleus, and in fact, it is active. It is turning on all kinds of genes. So that was consistent with what we had seen before. It looks like maybe FOXO possibly does have some kind of a role. At least it's in the nucleus in these models of diabetes. So then we hypothesized that maybe if we silence that, if we turn off FOXO, we could have some impact on diabetic cardiomyopathy. So this nomenclature may not be familiar to you, but you can put DNA sequences around a gene, so-called LOXP sites, and then bring along an enzyme called a recombinase, a Cre recombinase. It sees these 2 LOXP sites, and poof, pulls out what's in between them and silences whatever gene was in there, makes it go away and turns it off. That's called floxing a gene or silencing the gene this way. So we engineered mice where we floxed both the FOXO1 and the FOXO3 alleles. So they're floxed, they're normal, they're just fine, except when exposed to a Cre recombinase, and that's what C stands for here. This is, if you're not used to reading this, we expressed just in cardiac myocytes only, that's the only cell type we're working on, is cardiac myocytes, a Cre recombinase that is flanked by a modified estrogen receptor on both sides. So you put all this together and nothing happens, everybody's fine, except when you expose them to an estrogen analog, tamoxifen, suddenly the Cre comes off, finds these LOXP sites, boom, and silences those two genes. So that's called a double knockout, and we also engineered mice to just flox FOXO1 by itself. We gave these animals diabetes by exposing them to a high-fat diet, and they became obese. You can see all three genotypes are obese. They are glucose intolerant. The glucose tolerance test is abnormal. They're hyperinsulinemic and hypercholesterolemic. So these animals have diabetes. Now we have the opportunity, now that they've got diabetes, let's pull FOXO out of the nucleus and see what happens. So in that experiment, we took animals and exposed them over 6 months to a high-fat diet and tracked contractile performance by echo during that period of time. Under wild-type type conditions, we found that the heart function, the contractile function, declines precipitously over this period. This is diabetic cardiomyopathy. As a control, we also did that in the animals that only expressed the MER transgene, and you can see the very same thing happen. There's this progressive and dramatic decline in contractile performance in these animals exposed to a high-fat diet, which by the way is equivalent of a Big Mac, large fries, and large Coke for all the time for 6 months. So what happens if we take these animals and we silence FOXO1 and FOXO3 and then expose them to this phenotype, to this paradigm? You can see that the contractile performance is preserved in the double knockouts, allowing us to conclude that FOXOs are in fact required, both of them are required for the high-fat diet-induced cardiac dysfunction. What happens if we silence FOXO1 only? Same thing. We saw the same protection when animals are exposed, when the FOXO1 allele was silenced exclusively. So in other words, we found that this FOXO1, in fact, which is in the nucleus, is in fact contributing quite a bit to this progressive myopathic phenotype in the setting of a high-fat diet. Taking it down a notch in terms of mechanism, when glucose comes inside the cell, the first thing that happens, it is phosphorylated by hexokinase, and down it goes down the pathway. So if these animals, which are diabetic, you would expect that they can't use glucose, right? Because they are insulin resistant, hence the cell can't use glucose. What does FOXO do to that? So we track that by PET scanning. You can see the heart here is a robust utilizer of glucose, as you're aware. So using 18-fluorodeoxyglucose PET scanning, we're able to track the myocardium's ability to handle glucose. And we found that under normal conditions, the LV uses plenty of glucose, but in the setting of high-fat diet, it does not. That shouldn't be very surprising. These cells are insulin resistant, hence they can't bring in glucose. Those data are quantified here. On the other hand, if the animals are exposed to a high-fat diet and we knock out FOXO just in the cardiac myocytes, they continue to be able to handle glucose normally. So in data that we haven't published yet, a paper that we're asking 3 questions. If FOXO1 really is a bad actor in this disease, it ought to be activated very early, potentially before other events. And so to test that hypothesis, we exposed animals to a high-fat diet for just 2 weeks. And you can see the animals on the high-fat diet gain weight, they become hyperinsulinemic, and in fact, if you take cells and put them in culture and expose them to insulin, you can see the AKT lights up. The cells are able to respond, they're insulin sensitive. 2 weeks of high-fat diet, that insulin sensitivity is gone. It only takes 2 weeks of a high-fat diet to render those cardiac myocytes insulin resistant and with FOXO stuck in the nucleus. So early on, FOXO, even after just 2 weeks, the cells are already headed toward diabetes and FOXO is already in the nucleus doing its mischief. That occurs before anything else occurs. You can see the heart function is still normal. There's no abnormal gene expression. So it does look like that FOXO1 nuclear expression and gene translation is in fact very early in the pathogenesis of diabetic cardiomyopathy. The second question we asked is, I showed you before that we can prevent that myopathic phenotype by silencing FOXO. Is any of that reversible? Is there any of this the plasticity that I showed before in pressure overload, does that pertain here in any way? So we took animals and we exposed them to 6 months of high-fat diet, but we exposed them to tamoxifen. Remember, that's the molecule that allows us to recombine the allele and silence the gene only halfway into it. So the animals that were exposed to a normal chow diet, no matter the genotype, floxed, which is normal, mercury myrrh by itself. And so this is the combination you have to inactivate the gene. You can see the heart's just fine. On a chow diet, nothing happens. In animals that were exposed to high-fat diet, as a control, the FOXO1 floxed allele, again, like we saw before, cardiac function declines. As expected, since this allele is just floxed but not recombined. The mercury myrrh transgenic is the same. So here's the interesting question. What happens on a high-fat diet where at this point we turn around and we silence FOXO1 just in the cardiac myocytes, and we found this remarkable return of cardiac performance over the ensuing weeks to months, despite the presence of persistent exposure to high-fat diet. Not only do we see the return of contractile performance, but the hearts shrunk. The hearts went back toward normal. This myocyte hypertrophy diminished. The fibrotic response declined. Fibrosis is quite plastic, more plastic than sometimes we remember. The ability to use glucose, which went away, returned. And the steatotic response resolved. Amazing. It's just amazing that by just taking FOXO out of those myocytes, despite the persistent exposure to high-fat diet, many of these events reverse. It's quite remarkable. The final question I asked, and I'll go through this real quickly since this is deep stuff here, is how in the world does a transcription factor signal back to the cell and break the insulin cascade? At what point does that happen and how does that work? And if you'll take my word for it, it happens at the level of IRS1. That is the molecule, the weak link in the chain, which is degraded. And it turns out the way that happened, that we've teased out, is by means of two kinases, S6 kinase and junk. So two different kinases that are expressed, driven by a transcriptional target of FOXO. So FOXO turns on PTK4, which activates these two enzymes and breaks the signaling cascade. So back to our model. So I hope I've convinced you, again, going through a lot of stuff here quickly, that insulin resistance is driven by mTOR. Remember, mTOR activating is capable of targeting IRS1, and it contributes to this third killer of human beings. So finally, to wrap this up, how in the world, given that mTOR, this remarkable molecule, is clearly involved in all these cellular events that participate importantly in several different important disease processes, how can we control mTOR? Now, obviously, everyone in the room should be interested in ways in which we can manipulate mTOR. So Cindy Morales, a PhD student in the lab, spent several years in data that we recently published, asking the question, how in the world do HDACs govern this? HDACs, as I've shown you, using HDAC inhibitors, govern these events in several different ways. The first question she asked was, is mTOR involved? It's a logical question because mTOR is central to all those things. And in fact, she found that cell growth, this is leucine incorporation, is attenuated if you knock down, if you silence HDACs using a different molecule. Apicene is a different one than TSA. And that response, the molecule rapamycin, which targets mTOR, doesn't add anything more. So in other words, if you target HDACs or mTOR directly, you don't get any added benefit, suggesting that they go through the same pathway. And in fact, the activation of mTOR, this signal here is the activation of mTOR, is attenuated when you silence HDACs, suggesting these and other data that in fact mTOR is the target that the HDACs are conferring. That also pertain to a variety of other stresses, phenylephrine, endothelin, and so forth. if you knock down selected HDACs 1, 2, and 3, which are the ones that are the business end of all this, she found that you in fact have to knock down all 3 of them together to attenuate this response. So it does appear to be a bona fide histone deacetylase-driven event, because either pharmacological targeting or a genetic targeting strategy are similarly effective. And the HDAC-induced, the mTOR activation that occurs in thoracic aortic constriction is similarly responsive to HDACs. And in fact, if you engineer mice that are silenced for both HDAC 1 and 2, there is an attenuation of that response. It is not abolished, because we believe HDAC 3 is also involved. It's hard to make HDAC 1, 2, and 3 mice. But it does look like, in fact, HDAC 1 and 2 and 3 are the targets acting that govern mTOR. And so finally, there are about 20 different molecules that govern mTOR activity, and Cindy checked them all and found that the one that was the business end is tuberous sclerosis complex 2. It is a brake on HDACs, and she found that if you take that out of the equation, if you silence TSC2, all of that response is attenuated. So then, to summarize, just briefly, this was Cindy's PhD dissertation here, that HDACs are a brake on TSC2, which is also a brake on mTOR. So in other words, the way in which HDAC inhibitors, again, which are drugs that I've shown you, hopefully convincingly, participate in a wide array of cardiomyocyte biological events, are suppressors of a brake, which they brake on TSC2, which also is a brake. So in other words, here's our model that in some ways summarizes much of the work that my lab has done in the last number of years, that this remarkable molecule, mTOR, participates in several important cellular events, which clearly in preclinical models are important to a wide range of, I think, clinically significant disease phenotypes. And mTOR is in turn a novel pathway of mTOR dependent control driven by HDACs, where HDAC inhibitors blunt them. So I hope that wasn't too much basic science, but here's the group that I'm pleased to work with. Diane Cow, who I mentioned, did the early work with the pressure overloaders right here. She now has a K08 and a faculty position. Cindy, who did the work in the recent science paper, is right here. And that's my group. And with that, I'll be happy to answer any questions that I can. Yes, Anne-Marie.
So you mentioned that all your studies are on the heart, but in your ischemia reperfusion work, it seems like a lot of that could be translatable to the brain. Are you looking at stroke or the role of HTAC inhibition and reperfusion in cerebral?
What a great question. The answer is yes. We're doing that experiment right now in stroke. And you know, as you people know very well, the mortality from MI these days is pretty low in the West, but stroke is still a devastating thing. And so we're actually doing that experiment right now in a stroke model that we're ramping up. You know, it's surprising to me that the industry is just completely unwilling to consider cardioprotection and reperfusion injury, because they've been burned so many times, and they're just not going to go there. And so we're trying to use a different strategy. One is to do a STEMI trial in China, but the other is to look at a different disease like stroke. Yes, sir.
So that was terrific. Thanks. And even for a non-basic audience, I think it's great, the stories that you tell. It's really terrific. And you may know, we've actually done a lot of the work in myocardial protection trials here. We've done, and personally, actually, I've been involved with several major efforts of futility and failure. One place, the one, you know, I think there was probably a little bit of benefit from adenosine on clinically relevant myocardial protection, but it wasn't enough to be clinically important. The one place where there was a positive trial was with sodium-potassium exchange inhibitors, an epiroid, in the setting of high-risk bypass surgery. It was actually a really compelling, significant reduction in myocardial necrosis biomarker release. but it was accompanied by a off-target effect of increased stroke. But it's something to think about. It's actually a nice model because it's not perfect for MI because you can give the drug beforehand. But I think it is a nice clinical model of ischemia reperfusion where with a modest number of patients you can actually look for a kind of a biologic effect that's actually translated into some clinical benefit.
Yeah, that's a good thought. We've thought about that. I'm aware of the sodium hydrogen exchange studies. The injury at the time of bypass surgery is, at least our thinking was, that in order to see a benefit or conceivably a harm, you need a really big signal. And so our power calculations are that if the benefit in humans is half as much as we and others have seen in mice, rats, and rabbits, that you'd only need to study 100 mid to proximal LAD anterior wall MIs and answer the question. And Merck, who owns that drug, is not interested. It's coming off patent in a year. It's complicated, and so I haven't given up. But our thinking, right or wrong, was that we wanted a big signal so that we could, detect, either improvement or worsening like that. And there's a woman that I know in China. I mean, they have three quarters of a million STEMIs in China right now, going up and up and up. And she said, I could do your trial in a month in my hospital if the drug were approved in China.
Yeah, it's really fascinating when you talk about something like mTOR and you try to inhibit its activity. And how can you discern sort of the off-target effects, i.e. creating safety, when you're dealing with such a fundamental part of biology? So you must have thought a lot about how you deal with that.
So I have. So you're right. So mTOR is all over the place, right? And for example, in the reperfusion experiment, I'm not proposing to treat people chronically with an mTOR inhibitor. Just one dose at the time of reperfusion. Even Merck was fine with that. Everybody was fine with that. Just one dose at the time of a heart attack. In terms of FOXO, which clearly is a bad actor in diabetic heart disease, FOXO is in other cell types. It's in hepatocytes. It's in beta cells. So we would have to find a myocyte-specific way in which it is. I didn't tell you much about the fact that it's governed by its nuclear versus cytoplasmic location, moves in and out. And if we could find a myocyte-specific way in which that is controlled, to keep it out, for example, in myocytes, or to promote its being left out, that's the direction we're trying to go after. But you're absolutely right. Some of these events are near ubiquitous, or certainly are beyond just the cardiac myocyte, for sure. Yes, Bob.
I'd be curious your thoughts. So there's some of us in the heart transplant space that we use mTOR inhibitors and modulate that system on a daily basis, and clearly that's a kind of distinct patient population, smaller overall, but wondering how your research in the implications for immune suppression and kind of that overlap of heart failure MI versus transplant population where we're using this for coronary artery vasculopathy and other implications.
Yeah, so to the extent that we will often check our results against rapamycin, we see the same sort of thing, that these clearly are implicating mTOR, but as you know very well, the off-target effects of rapamycin are legion, and so for that reason, we're not considering a chronic mTOR therapy at this time, but rather a surgical shot at one point. I have said that I have thought about patients with HFpEF. We've not done this experiment, but my hypothesis is if you take someone with severe LVH and treat it with an HDAC inhibitor over the course of 6 or 12 months, you could sculpt their LV back toward more normal size, and who knows, maybe have an impact on HFpEF. That would be a situation. That's the farthest I've gone in my thinking. Is maybe 6 months of this with monthly echoes to see how you're nibbling away and remodeling the left ventricle. But that hasn't gone beyond the thought experiment at this point.
this is kind of a little bit tangential, maybe, Joe, but I heard this great lecture from your team member Darren McGuire about bariatric surgery and the early effects of bariatric surgery on kind of metabolic changes in diabetes. And is anybody using that kind of as a model to explore kind of modulation of diabetes and its effect on the heart that you're aware of?
I'm not aware of that. Like a preclinical model to try and understand how bariatric surgery, weight loss induced by bariatric surgery is, clearly it's beneficial and incredibly rapidly so, right? And yeah, it is. That's my understanding.
Like the gut is maybe a lot more of an endocrine organ than we'd realized, it seems.
I think that's exactly right. In fact, they have this sleeve now where basically the food comes out of the stomach and bypasses the duodenum and is dumped out, in the distal ileum, and that makes a huge difference as well. So people have localized it to certain parts of the gut where those events are occurring, and there's a lot of work in the gut microbiome as well that high-fat diet-induced changes in the spectrum of bugs in your gut. There's some evidence to suggest that too leads to, for example, an inflammatory response. Lipopolysaccharide levels go up all over the place, and a toxic effect on the heart from that. Yes.
So, given your findings with the sort of epigenetics of heart disease, do you think that there's things that happen sort of early in life, or exposures and things like that maybe play a big role? So, for example, if you have diabetes and obesity early on, and it's corrected, is that going to play a role in heart disease?
For sure it does. Yeah, like if your mother was obese while you were in utero, that has an effect down the road. These things are starting to crystallize out, but there's strong evidence to support the notion that in your ancestors, their exposures to a variety of environmental cues triggers, cascades down to you through these epigenetic marks, for sure.
Great. So thank you very much, Joe. That was really a great expose.
My pleasure. Thank you.
And I love the vegetable analogy.
And so now the rest of the year is going to be much easier.
Yeah, on to the beef. Thank you.
My pleasure.